Shall we start?
My mic is audible, right? Yeah.
Okay. So, good afternoon. Welcome to this second episode of Lab Rats and Field Mice. Myself, Bivas Nag, a co-committee member of Theory Club, welcome you all to this talk, which will be given by Omkar Khache on the niche partitioning in bats. Talking about Theory Club, Theory Club is a student created club supported by the Wellness Center where we focus to cater, communicate, and discuss different theories in the form of stories, talks, games, movies, and other medias. So, today in the second episodes of Lab Rats and Field Mice, we have with us Omkar Khache, who has done an internship under Professor Rohit Chakravarti, and the study was named Niche Partitioning in Bats. To begin with, I want to introduce our today's speaker, Omkar Khache, who is pursuing a master degree in botanical sciences from St. Xavier's College, Mumbai. He has varied range of interest ranging from ecology, plant biology to evolution. He is also an ardent supporter and follower of the great Sir Charles Darwin. So, from the Theory Club, we welcome you, Omkar.
Thank you.
So before starting this, this is a very pretty interesting internship or project you have done. My first question will be like, what motivated you or what drove you to choose this project, and how you got to know about this project?
Okay, so I think I'll answer the latter part of the question first about how I came to know about this. I think most zoology botany undergrads might know about this mailing group called Yeti, which is Young Ecologists Talk and Interact. So they normally send mails about like any positions if it's open, like an internship position or like a PhD postdoc around India, many times outside the world too. Whichever researcher needs someone to work with him and all. So I'm a part of the mailing list, and I saw this mail when I was just looking out for internships, and it seemed pretty interesting, and I thought I'll go for it. And answering the first part about motivation, I mean, I had a motivation to do a good internship. I mean, this was in my gap year, and I wanted, so after giving all my exams, which I took the gap year for, I had a lot of time, and I thought I'd get a good internship done where I can learn about stuff. So, and this mail came, and I didn't know anything about bats. And I wasn't looking for any specific internship about bats. I just wanted an internship about ecology, any area that spans ecology and evolution, and something I learned. So this internship came by. It was about bats, and I bats always are very mysterious and interesting. To some people, scary. It's fine. Yeah. So yeah, it seemed pretty interesting. And on top of that, was in Uttarakhand. I've never been to Uttarakhand, so that way was quite interesting. And like hiking and going up. So yeah, and also it was an excuse like for a mini road trip. So I took my bike there. So all of that came together into this internship.
Great, great. That's great. And as you know, as you talked about bats, I have a very intriguing questions in my mind revolving around that. Have you asked this question, or has this question that's spurred in your mind, that why bats? What is the ecological significance of the bat? Like how bat is important to our environment of the ecological system?
Um, yeah, one thing is bats are really important pollinators. I mean, some flowers are exclusively pollinated by bats, mostly fruit bats. The big one that you see flying is like the Indian flying fox, but the other smaller ones that normally pollinate many of the flowers. And it also is a major seed dispersal agent. Like most of the figs you see, there are a lot of bats around fig trees. So that way, even the Ashoka trees, if you have ever wondered why do they keep growing here and there, it's because bats. If you can see them eating the fruits here, if you I don't look. So yeah, my mainly seed dispersal agents. These are the fruit bats, and most of the insectivorous bats eat a lot of moths and other arthropods. Might keep their population in check and all. That's it. Yeah.
That's great. That's pretty interesting. Coming to the name of the study, like name of the study seems very appealing, like niche partitioning in bats. So is niche something related to habitat, or how you describe niche, or rather how you describe the topic of your study?
Okay. So what? Also, another thing, Rohit is actually a PhD student who is still. I think he will take interns next year also. So kind of look out for his email near March. It's an amazing place, an amazing internship. Other than that, yeah, about niche partitioning. So like for example, so you have a habitat, let's say a forest habitat or like any kind of forest that comprises of the habitat. And so a habitat is not uniform. It's not uniform always. In the habitat, spatially some parts are different than the other, and some organisms specialize in a specific sort of micro habitat, if you might call it, and that's called the niche. So what niche partitioning, let's say in the context of bats, means that, let's say if I take a forest ecosystem, and the different sort of micro habitats would be, let's say, upper story regions, just above the canopy, in the forest parts, probably a stream that runs through a forest. So those are some of the niches that different communities of bats exist in. So different species compete in one habitat using different niches, and that kind of makes it efficient, the competition and the way the bats exploit resources. So
That's great. That's great. Like
yeah.
as we talked about niche being a micro environment in a particular habitat where uh, different types of bats occupy in a particular, like in a particular habitat. Let's say for the example of tree. So like when you were studying on niche partitioning of bats, what was the motive behind this? Like what was the expected result that you were expecting out from this study?
Yeah, I think more than it, this was like a, so I mean, this wasn't like a hypothesis driven. I don't think this was a hypothesis driven research. It was more like documenting it. Kind of, it was like a primary research where you see what's going on, and based on the data you have, you try to correlate different variables and try to, see a pattern or a trend, and that. So yeah, so I think he was looking at different bat communities in three niches. One was forest, inside the forest, like forested region, forest clearing, and forest edge. So these were three niches inside the forest habitat, and we were looking how the bat community changes inside these three niches in several different altitude regions. So yeah, so we just wanted to see how the bat community is spread out in different niches, which bat occupies or which bat species occupies a specific niche as opposed to other. Do some of the bats flexibly use different niches in the same habitat, and then correlate it with the call data that we gathered and the size of the bat and other features of the bat. So yeah.
That's great. That's pretty good work, I think. And when you talk about like correlating different data, so correlating different inputs that you get from different observation. So what was your approach for this study, or what were the methods, or what were the steps taken by you, or most appropriately, what are the safety measures? Because like working with bats in Uttarakhand, you have to work at night, so there are like many risk issues as well. So what are the safety issues that you need to take, and as well as the steps that you need to take
Okay, so
during
yeah,
this course?
I'll answer the safety things first because it's a small answer. And okay, so I think the most safest we had were gloves to handle bats because some of them can bite. Like some of the bigger ones that we collected, bigger as in they were all kind of under 60 grams. The highest was 60 grams. Most of them were like 15 grams, 7 grams. Some of them were like 5 grams. They're quite small. But Yeah, one was gloves. Just if you're comfortable not wearing gloves, that's fine because gloves kind of restricts how you handle the bat, the movement and all. But other was having a rabies injection before you go there, taking 3 rabies shots. Other than that, there were no safety measures as such. Just stay in a group because it's a black bear territory and you don't know what can happen. But yeah, it was pretty chill. It wasn't really unsafe.
Great, great, great. So as you talk about your study and the internship, can you tell me some of the like things that you did in internship? Like, as you mentioned, like how you collected the data. So what are the type of data that you collected?
Uh, so, so first thing, so again, as I mentioned, we were collecting data in one altitude region in three different niches that I mentioned: forested, forest clearing, and forest edge. So what we did was before we misnetted at the place, we went and set detectors there, which are passive detectors. So basically, what passive detectors are, they're automated and they're programmed so they switch on at a particular time in the night and switch off at a particular time. So they used to be set off at dusk until 5 o'clock or something. They used to be switched off. So these data collectors just record the acoustic activity in that specific region. There's a mic connected to it and it records the specific activity in that region at the time they switch on and off in that particular. So we set these first at the places we messnetted in 2 days before we messnetted at these different sites. And then the next part was messnetting at these sites. Messnets are nothing but they're basically really thin nets that people also use for bird studies. And these thin, they call mist nets because obviously you can't really see them unless you're quite close. So you hook these nets up on like poles. Most of the nets are what 6 meters, 3 meters, 9 meters. Depending on where we were mist netting, we use these nets. You string them across a forest path or a stream or something, and you wait. So that was the normal. That's how we captured bats. Then the next part is we collected morphometric data of the bats. Morphometric data is basically just measuring the wing length, I mean the wingspan, measuring the height, measuring the fore length and all, measuring the jaw length and all that from a specific molar, or I forgot which exactly, but yeah, and the weight of the bat. So these are morphometric measurements we took, and then we captured the picture of the bat wings and all. And now the next part was actually also part of the study was diet analysis, which was done by taking a tissue sample, small, really small circular bat wing, and also a blood sample and capillaries, and we put them in small Eppendorf tubes. This was what a routine day was when we misnetted. Yep.
Okay, so as we talk about like doing the diet analysis and also other things like weighing the bat. So what was your size of the study? Like when we are doing a field work, you need to have a size of study. Like how many bats you did or how many bats data you collected during your tenure of the internship. And in addition to that, I want to ask, like you also mentioned, and as a poster of the talk suggests that you are also capturing some of the passive recordings of the bat. So if you can elaborate on that as well.
Okay. About the sample size, I'm not sure because, I mean, Rohit is designing the field of study, so I mean, he would be better one to answer that. But it's really variable. Like you can't. The thing about capturing bats, like what I saw, you can't really. I mean, you will have a sample size in mind when you go into the field and do your research. But it's very variable. Some nights we never caught a bat. Like we literally never caught a bat. Some nights we caught like 30 bat individuals, like 30 individuals basically, and some nights two. So it really depends where we are misnetting, and that way it's variable. Then you asked about passive recording. So like again, like birds, bats, like obviously you can't hear bat calls because they are in frequency way beyond what a human ears can comprehend. They are above to 20,000 hertz. Many bat calls are like they start from 15. Like the free tail bat, we could kind of hear those clicks, like really high frequency clicks because it was 15,000. So most of the bats that we caught were in like 50,000, 50K hertz or like 80,000 hertz and also way beyond the hearing. So what these passive recordings do is they just measure all the bad calls. They triggered. There are a few parameters that you have to set so can they can get triggered at a specific frequency or intensity of the call, and they measure basically they record all the call activity in a normal SD card, the camera SD card, and then you analyze the call activity by using a software. You get a spectrogram like frequency versus
You might be
time graph.
using Audacity or something sort of that, like sound analyzing
Yeah,
software.
yeah, Audacity also you can use. He used Raven Pro. That's like a Cornell in
Yeah.
university software. Yeah.
Yeah.
So you get a spectrogram, and so looking at the spectral behavior of the call, there's the frequency of the call and the shape of the call, very well pinpoint to what bat species it can be, because every bat has a characteristic call frequency, intensity, and a call shape. So that's what, and also it gives you kind of the activity around that specific niche by looking at how frequent the calls are, who's calling in the recording, which bat it is, and you can do a lot of different statistical analysis to see, looking at the abundance of a specific bat, or how many bats are calling, what is it doing. So yeah,
Okay,
that
that's pretty interesting. Omkar, pertaining to what you explained about passive recordings, like I was working on Magpie, where I also recorded some of the vocalizations or acoustics of the magpie birds. So what I observed over there is like birds have different type of calls. Like when there is a particular prey predator, they have a different type of call. When they have a like they call for mating, they have a different type of call. So do bats also show this type of patterns of calling?
yeah, they do. Okay, so if so, I won't. So obviously we can't hear bat calls. We don't know exactly how they sound. So you just kind of imagine a frequency versus time graph. I don't know. Do you want me to draw it somewhere? I don't know. But okay, so
No,
I'll
you can explain. You can explain. Yeah.
yeah. So just imagine a frequency versus time graph. You have frequency on the y-axis and you have times in milliseconds in on your x-axis, because bat calls are really short and they are like very abrupt pulses. So many of the bat calls, okay, so many of the bat calls, so there are different types of bat calls. One is when it navigates, just kind of knowing about its surrounding. I mean, like say you're a bat and you're flying, and because obviously you use the auditory senses to know what's around you, just normally looking at around for obstacles, and normally looking at your flight path, just search calls that, and you get the echoes back, and those echoes tell you the range, how far the obstacle is, or what your flight path will be, from where will you be flying, and all. You get the information about the surroundings. So those are the normal search calls. It, it, I mean, they emit it at a uniform rate many times, but it's not very frequent. They emit it. If you look at the graph, you get these kind of distributed pulses on the graph. So one is a search call, which are high pitched call, because again a call needs to be loud and have high energy to get a good echo. So one is a search phase call. Second is the call when it kind of hones in on a prey. So whenever a bat locates a prey and it's just kind of flying towards it and going to feed on it, there's a thing called a feeding buzz.
Okay.
So on the graph, I wish I could draw it. I don't know, just kind of search it after you go, whoever is interested. But feeding buzz is basically, those calls are really closely spaced, and they're basically like tuck, 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 tuck. It's like the bat is calling with a very high frequency of calls, many calls in the one particular time before it goes and catches the prey. And the third type of vocalizing a bat uses is a social communication. And these calls are normally long and they differ time wise or frequency wise, because, most social calls you have to get a meaning across. So they will be substantially longer than the search phase calls and all. They will have a lot of variation in the frequency, a lot of variation in the time and intensity which would they call. So yeah.
Okay, that's pretty interesting, and that's pretty like like when you put the calls in a Audacity or any software of that,
Yeah,
you can
you might have seen that. Yeah.
yeah, you can easily predict that what are the changes and what are the things that are going on. Moving to that, like I have a question. Like, bats are basically a social animal. They live in groups. So, is there any
Yeah.
behavior pertaining to that? Is there any behavior that birds show as it lives in a society, like any societal role that a particular bat or an individual bat is playing in that society? Because when we see it in the if I'm relating it with the ants, colonizing of ants, they also play different roles. So do this type of thing, or if you see in the case of bees, they also play different type of roles. Do these type of hierarchies are present in the bats?
I'm not very sure, but I'm sure. I mean, many times when you see some of the bats when they fly with their pups, so the nursing and parental care aspect is there in bats, and obviously they live in like huge colonies in caves and other sort of nest roosting areas. But I only know one example about a vampire bat, which it nurses the young of even other than its own. It nurses offspring other than its own. So yeah, that's all I know. I don't know much about their social behavior. Yeah.
Okay, that's pretty cool. That's pretty cool behavior, I guess. When you talked about the diet analysis, so what were you looking for when you cut out the tissues and put it on a pen off, or cut out the tissues from the wings or anything? What were you looking? What were the What were the measuring criteria? What were you looking for when you took the tissues?
Okay.
Because
So
when we are
yeah.
doing a particular experiment, there should be a like set of expectation that we are expecting.
So I think the diet. I mean, obviously most of the bats that we caught were insectivorous. Like most echolocating bats are insectivorous or eat arthropods. So like here, when we took the tissue samples, we're basically looking. They were going to do a stable isotope analysis on the tissue sample. So now what is stable isotope analysis? Stable, so I mean many elements, like basic elements that make up food or energy like carbon, hydrogen, nitrogen, yeah, carbon, hydrogen, nitrogen, have isotopes, like elements, I mean they have kind of, how do you call it?
I am
It's the same element but at a different
I
higher
am
mass. Yeah, exactly. Yeah. So looking at the profiles of these isotopes in the tissues, so, and you kind of can make an analysis of what the bat might have eaten, looking at the profiles of the ratio of different isotopes present. Because after whatever the bat's going to eat is going to get assimilated in the tissues and the blood. So by looking at the ratio of different isotopes in the blood, you can compare and kind of predict, not obviously to the level of species what it has eaten, but the kind of food composition or what it might have eaten, looking at the ratio of all the isotopes present in the tissues and all.
That's very interesting because like we do in the lab like various type of analysis, like you take samples from gut and all, but here you're comparing and looking at in tissues and comparing with the isotopes, and I think that's very much interesting. Now coming to a generalized question that I want to ask you is like like working with bats, you have to work at night, and when you're working at night and at a place in Uttarakhand, and field trips always looks interesting from the person who is looking at it, but there might be some difficulty that you face while doing field trips, like it may be very much tiring. What are the pros and cons of doing a field trip or doing a field internship, apart from like doing a lab work or wet lab techniques?
Okay. Another thing, I don't think it's more like pros and cons. It's more like what your question demands. I mean, if you're working on something that's a non-model organism, or you're working on with biogeography, obviously there's a lab component to it. But yeah, I think the different challenges when it comes to field and the lab, but I think I would like to highlight what are the similarities when it comes to both. I think in both of them, you really need to be organized in one. Like, you need to have a good sampling design, kind of, tweak it so it's constant. You need to organize the time. And I think when you kind of start doing your own project, you kind of know how you organize the time with, for the lab, it would be just making your reagents and, before making a culture beforehand, keeping the autoclave ready and all that. For field, it's more like changing the batteries, keeping the batteries charged for the detectors, keeping the poles organized, all the bags in place. So that part is common to both lab and field. You need to be organized, constantly tweak things, look out for improvisation and all. Okay, so with the cons of fieldwork, or fieldwork is obviously very interesting because I mean you're out in the open, but it's not always that rose, like it's all rosy, you're out in the open, like how they advertise, wind in your hair and all. It's not. It's more like sweat, grime. At times, even like shit, I want to go back to the base. It's too cold. Oh shit, my shoes are wet. Oh fuck, leech. Is it fuck all right? You can beep it later, but yeah. So that I think so. Yeah, what difficulties we had during the field was, I mean, it was very tiring because Uttarakhand is a hilly region. Even if you walk 1 kilometer, your field site is just like a few hundred meters, 1 kilometer away. It's a climb many times. Like 2 kilometers away, we literally had to climb with some poles and bags and all, and with misnets up there. And it wasn't like I'm as difficult as I'm saying, but yeah, it was tiring. If you're not used to climbing, it is quite tiring. Thing is, just because you have to climb every time, and climbing in cold, the air is slightly thinner there. So, you're like, every like few hundred meters you stop and just take a huge breath. So yeah, that bit was tiring. And the second was many times the nights were cold. We just have to get used to it, thoda. The worst part was, like it was so many times we netted across streams, and like the bats were caught, like say bats were caught mid net. You literally had to get down in the stream ankle deep water, slightly deep, and the stream and the Himalayan streams are so goddamn cold, and it's like 8 o'clock, and the bat is to be entangled. It's not that the bat is in a net nicely lying. You kind of have to untangle it, and they move a lot, and they get entangled into knots, and also you have to be really careful. It takes 5 minutes at them to just remove one bat from the net, really untangle, and all the time you have to be in that stream. Literally, your legs are like numb, but you have to get the bat out because it's just kind of sad seeing the bat distress in the net, entangle. You have to get its wings and all the digits kind of untangled from the net. So that the cold water was obviously a huge thing. Yeah, and many times it used to rain. So,
Yeah,
and I
and
fell,
also
yeah, go on.
yeah, and also like I want to, like, as you said, as you mentioned earlier, like you talked about, like when I talked to you about the sample size, you so said that one day you got 30 and one day you got 2. I think that also a bit problem that you face,
Yeah,
or
yeah. I mean, I think with all field studies, the thing with lab studies, you can select the variables that you want, and you can keep other things constant, and that's one huge boon of lab work. Like that, your head is at peace with the variables, and you can look at things, and it's more systematic. The thing with field is you just have so many things not in your control, and it can get frustrating, especially the weather. And then you start literally abusing the weather after a point. You're like, why do you have to rain now? Like, why? And it's just so the weather is a more. I remember seeing Rohit get so frustrated at weather, and then I kind of understood him because yeah, the weather literally, you just nicely set nets up and then it rains, and you're like shit, and all the leaves get stuck in the net. So instead of removing all the bats, you're removing the leaves from the net. So yeah, that way variables wise lab field work is slightly messy and more of a headache. But you have to improvise always here and there.
yeah, that's the thing, that's we need to adapt, or we need to acclimatize ourselves according to the situation.
Yeah.
Okay, so moving forward, I want to ask, like, field works, as you said, there are many like ups and downs you get in regular places, and there are many memories that you create. And of course, doing an internship in Uttarakhand, as you said, you carried your bike as well along with you. What are the memories that you want to keep it for with you, like after this field study?
So I think
Like, what are the
I'll start from the
different
start. I
instances that
think
you
I
want
was
to highlight?
always just when I entered Uttarakhand, I'm like, when am I going to see snow-capped mountains? And I was riding, and then one turn I saw those snow-capped, and I literally stopped. And I was like, okay, this is amazing. I think as soon as I entered Uttarakhand and the mountains began, I just was taking pictures because I don't know, there's something different about Himalayan streams. They're like green and it looks so amazing. But yeah, I think so ups will be just the site, like literally where we were staying, we could see the top of a place called, I don't know if I forgot what the Rudranath. It's like a temple up there on the height, and we could see the mountain. The view was amazing from where we were staying itself. So view wise, we got used to it soon. So like all the just look landscape wise, field works amazing. We saw amazing things, and yeah, a lot of shit also happens. Like I remember falling in the same stream twice, like 3 weeks apart at the same place. I was taking the net across the stream and literally fell in the stream twice, and I had to do the rest of the field work in all wet clothes in the cold. And yeah, and on top of that, chaffing also because you're wet. It's irritating. So yeah, like that. Oh, yeah, it's a lot of stuff. Many times, once I tripped over the net, and with me, I took the bats down. I literally had to take one hand, tie the net again up straight, take the bats out, all of them, keep them in the bag. So a lot of stuff happened. Like, I mean, you have a plan, but you go in with the whole idea the plan is going to be screwed. So you kind of be ready to get upset and improvise. It's fun. I mean, in a way, if you look back on it.
So here, plan A doesn't work. You always have to keep plan B, plan C, plan D ready.
Yeah, yeah, more than yeah, just kind of do things out there. Like you have, obviously you should have past experience. You have read about other people doing it. Just have to keep that improvising ready till you find a nicely stable sort of sampling method. So
Okay.
yeah,
So talking about your internship a little bit in more detail, like this internship was a group internship, I suppose. Uh, yeah. So how was work? How was you or how you felt when you were working in a group? Like how was the members in your group, and how was the professor who was guiding you?
yeah.
What are the memories with them that you created, or what are the instances that happened that you were in part of? Any discussion, any constructive discussion, any constructive, like helping each other, knowing each other, you made friends. What was the experience out there working in a
So
group?
we so Rohit had two local volunteers. I think they're local people. They were Gujars from like Haridwar and all, which is slightly down from Garhwal where we were. And they normally were his long-term volunteers types, those. And I was like the new one he was taking. They are there almost every year with him. So we were like group of 4 people, constant. A few people went and came. A guy from Delhi had gone for a few days. But yeah, we were constant 4 people. I think start you are just like kind of skeptical about everything. Like who am I working with? What is it going to be? It's like all curious. How are they going to be like? And things just level out after a while, and it's nice. I mean, and Rohit is amazing. Like working under him is really cool. I was at one internship in IIC, and my whoever I was working under was so non-approachable and such a pain. But Rohit was amazing. He lets you do stuff, and then you feel kind of valuable as an intern because one of the most daunting thing as an intern is feeling useless and being like this work can be done by others. Why am I there? But he lets you do work. He makes you feel responsible. He let me handle baths really early in the time, even if that was data. So that was really cool. So like that, it was amazing. Then you bond over because you are together like for 2 months straight at different places. So many memories created to tease each other. All that stuff goes on. The most and we were the most competitive was playing Ludo because in the morning we had no work. Most of the back work is at night unless you're setting detectors up. So we played a huge amount of Ludo. And it was so competitive, quite competitive. Yeah, it's just nice. A lot of people, fun.
That's great. That's pretty great. And
Yeah,
like working in a group is always amazing because at every moment there's something or the other goes on, and one supporting other supporting, and like create memories, small talks that you remember for, and this all adds up to your internship, or rather it gives you an overall experience of doing a work. Okay, now moving on, like you worked on bats and you love to work in ecology field or like doing a field trip. What are you planning for your future? Like what are the things that you're looking for, or any opportunities that you're looking for to work on?
so I mean, future would be so. I think future, yeah, so academic person, I don't know, but yeah, I think PhD would be the natural step to take. But I would still want to get more experience working on projects So I would really want to be like a research assistant or like a junior research fellow somewhere, because I
That's
think
great.
I still need to be more independent in terms of, yeah. I'm doing my own project. That's what I'm doing right now. But yeah, let's see.
That's great. That's great. And being, I feel like what my take on field thing is field trips is like being independent gives you a like lot of faith on yourself that you can move with a plan and you can go ahead, and if you fail with plan A, you have a plan B, which is made by you only. So you need not have a support of another people, but you can move it on yourself. Uh, Talking about the current scenario with the future, what is the future planning, or what is the future planning of this study? As you know, there's a lot of scientists who have quoted that there's a relation of COVID, or the coronavirus that is causing COVID is being originated from the bats. So what is the future of this study that you have begun with? And I think you have done a pretty good job, and what you are seeing, or what you have, or what your professor is planning to do after this amid lockdown due to the coronavirus, and it's being like many scientists are speculating that it's being originated from the COVID.
Yeah,
It's originating from the
I
bats.
mean, it bats are like the likely, but they also said that there might be another agent between bats and humans. They said snake or pangolin. But anyway, I mean, yeah. Okay, it happened. It was like a rare thing, where shit goes wrong. It happened. I think, I don't know, shouldn't really panic, because even, I know also coronaviruses, also civets harbor coronaviruses, and also raccoon dogs. People still love their civet poop coffee, so I don't know. Yeah. So regardless, I think there would be more research into coronaviruses and bats and their relation. But I also think, I don't know, forest officials and local communities will be kind of more deterrent about researchers working on bats close by. Because, like, in Uttarakhand, when we worked, Rohit had really good relationships with the local people there. Like, they knew we were working on bats for years, and we were in good terms with them. They respected us. They called us to the weddings and all. We literally had so much free food at the weddings. And so, like that, but I don't know how comfortable they will be now, considering all the COVID, and people get scared, blow it out of proportion and all. And already bats are not as popular as any mammals are. I mean, there are a lot of mysteries and blah blah witch tales and shit about bats. Common in local communities. So yeah.
Okay, there's always an uncertainty after this amid lockdown, and then like field research has been like sort of went through a pause stage during this lockdown,
I think
and
things will die down. I mean, things will return back to normal. People don't hold things for long. Everything will be fine.
okay, so I will be taking some of the audience questions that has been like
People have
coming
asked
up on
now.
the stream.
Actually.
Yeah, there are many questions.
Oh, okay.
So first question is from Navneet Mishra.
I know.
Is there any relation between call frequency and body size of the bat?
Ah, I think also, I don't know, but I'm not sure about that. But many of the horseshoe bats call it really great frequency. I think frequency is more related with the kind of surrounding the bat is foraging and operating in, the kind of prey it's looking for. Because frequency is going to give you the frequency with which the bat calls is it's going to give you the resolution of the echo, what kind of echo you're going to get. Or it also depends on the foraging style of the bat. So I don't know, there might be a relationship, but also there's a relationship between the frequency of the bat calls and which surrounding is operating. And some of them can modulate the frequency they call in depending on how cluttered or open the surrounding is.
Okay, that's pretty much answered Navneet Mishra's question. Another question is from Gunu Patak. I heard that bat skulls affecting the way they ecolocate. Can you tell me more?
I am sorry I can't, because I've not really read about it. They probably can, I think. I don't know. I wish I could answer that.
But can you tell Gunu some sites that she can go or refer to that sites? Anything?
I think I think you should just open Google Scholar and type bat skulls. I think you'll get a host of research papers which can help you. Or oh wait, there's a book called Bat Acoustics. Forgot the name of the book. I have it open here. I close it. But there's a book called Bat Acoustics by Griffin, something in Griffin. I think you should check that. That has all the neurobiological and other physiological aspect of what bat calls.
Okay. I think Gunu, you can check that book out. What Omkar has mentioned, bad acoustics. There's another question from Hira Shinde. Do all bad?
Actually, bad bioacoustics. Anyway, go on.
Yeah.
Yeah.
There's another question from Hira Shinde. Do all bats use sound to search for food?
No, only the bats are divided into megachiropterans and micro, which basically means big megabats and microbats. Most of the small mammals that in the microchiroptera are the ones that echolocate and hunt prey. The bats that are the fruit bats in the mega bats, they are mostly they locate their food by they're basically frugivores. They eat fruits and pollinate. So they base not pollinate actually nectar. They eat fruits and nectar.
Yeah.
Yeah. So they use olfaction, that is a smell, and they use vision to locate their food. Except there's a bat called rosettes, which is in the old world fruit bats, which actually uses slightly different sort of echolocation. It produces ultrasonic waves from its tongue clicks. So it clicks its tongue and produces ultrasonic, a sonar sound. Well, most of the normal echolocation, the small bats, insectivorous use the larynx to produce the echolocation signal.
And also, there's another question from Hira Shinde only. Like, do there any difference in the sounds of the bat, which feed on fruits and like frugivorous bats and insectivorous bats? There is difference in the sound they use.
I think frugivore bats will most likely use vocalization as sort of a distress call or something. I mean, have you ever heard huge as ruckus created by fruit bats under their colony on trees, like all the screeches and all? So yeah, I think the role of the vocalization will differ in the bats that hunt insects and fruits. The most of the vocalization will be to navigate the surroundings and hone in on the prey. Well, bats that eat fruits are mostly the vocalizations will be some sort of a social or a distress call.
Okay, that's pretty much answers Hira's question. There's a question from Neil as well. Do bats use navigation while they're navigating in the cities, or do they face any issues while navigating in the cities which have a huge buildings, like all the tall buildings out there?
I haven't looked at it, but looking, let's say if you're worried about fruit bats flying, shouldn't really. If you can, I think if you can successfully navigate into a huge ass thicket or a clutter, city shouldn't be a problem, except in case if sound creates a problem, like the din of the vehicles and just the city sounds creates a problem for the ones that use sonar. That they did a study with loudspeakers on bats. They said above a certain threshold it affects bats, but otherwise it really doesn't. Also, you have to remember the bats are working in frequencies that are like outside what we normally hear. So I don't know.
So like ecolocation is like it is a I think it's a budding topic where you can work on like a lot of work need to be done on urban ecology, like how this urban ecology
Yeah, it could be
could be done,
interesting, especially
yeah, affecting the birds and other species. Because I remembered in my studies when I was doing Magpie, I was using Magpie as a study model for the vocalization. We designed the study of using vocalization of Magpie uh, as a trait to mark the noise pollution.
ah, okay.
So I think a lot of work need to be done on the like
Lot of
effect of
yeah.
urban ecology, the development, our development in the ecology. Coming to the another question from Gunu Patak itself. Uh, do they use their throats to make the sounds, or do they use tongues and lips, or both, like we do change sound we do to change the sounds?
So most of the echolocating bats, the insectivorous, use a larynx, basically throat, I think that she means, to produce a sound, and they emit the sound either via the nostrils, or some of them, most of them emit via the mouth, and some of them also emit the sound created in the laryngeal space via the nostrils. And except that there's one fruit bat that I mentioned called Egyptian fruit bat rosetus, which uses its tongue flicks. It's not laryngeal. It uses its tongue, probably makes a motion with its tongue that produces the high pitched ultrasonic sounds. Yeah.
Okay, so another question, I think it's the last question. I got it from Hira Shinde. It will be interesting to study the neuroscience of deaf bats and how they navigate it.
Oh yeah, it will be also probably altering the ear characteristic of the bars. I don't know. I think some of them, I think back in the day, one of the Italian scientists blindfolded the bats and saw if they again come back to their roost, and they did, and that's how they figured out that they use echolocation or some sort of a sound. Try to ear plug the bats and see if they can still navigate. So yeah, that way it can be very interesting, I guess.
Okay, okay. So I think it was nice talking to you, Omkar. To conclude this session, I would like to ask you, like to request you to tell our viewers about the how you got the opportunity, or what you talked about Yeti Foundation, I guess. A bit more about Yeti Foundation, where they can look out for the internships like this, and any views that you want to share with our viewers, and you want to communicate something. Go ahead.
So first I'll talk about the Yeti. Okay, so Yeti is basically, as I mentioned, young ecologists talk and interact. The abbreviation is Yeti. So it's a kind of a foundation, if you might say. It tells the other ecologists, students, and whoever is seeking out for experience as a volunteer or something in wildlife. It gives you postdoc positions, PhD positions, whichever are open. Tells you about conferences held around you on the city, which city the conferences are held. Any ecology related topic openings, it normally tells you, mails you about. So what you do is you go to the site and add yourself in the mailing list, kind of subscribe, yeah, and you get the mails about things. Yeah.
Okay, that's great. That's great. So Omkar, it was very good talking about you. I hope like you carry out doing more research and more uh, exciting projects like this bats things and other things that you are already doing and being a part of it. Uh, so it's a warm-hearted welcome for you from the Theory Club to accept this request. And to conclude, I would also like to tell our viewers that our Instagram page, and we are always open. We have an Instagram page where you can check out and apply for this lab rats and field mice, which is a platform to give the students to discuss about the research and the internship they have been part of. And keep following us on the Theory Club YouTube channel, Theory Club Instagram page. We will be coming up with talks and more interesting researches that we have been working on. And I hope you have enjoyed the session. And Omkar has given us such great information that we can go and look out for, and always we can reach out to Omkar whenever we have any doubts or something. So thank you for listening to us, and I hope this was a lovely meeting that you got, and a great experience that Omkar shared with us. So thank you and have a nice day.
Thank you for hosting me.